So in the last lecture, I looked at other evolutionary mechanisms associated with disruptions of Hardy-Weinberg equilibrium, including non-random mating. And the last topic that we covered was the idea of co-adapted gene complexes, or the idea that a trait, an adaptive trait, may only be formed when you have specific combinations of alleles at multiple loci. And so a much more complex view of adaptive evolution compared to like the single gene models that I've been talking about in the past. So this lecture is going to focus on the concept of linkage disequilibrium, which is a mechanism to preserve co-adapted gene complexes once they form. Paradoxically, though, linkage disequilibrium can be a barrier to the production of co-adapted gene complexes. So the topic of linkage disequilibrium is going to be our introduction to how multiple loci can affect each other in the evolution of a trait. When we have loci evolving independently, we have what's called linkage equilibrium. And linkage equilibrium really is kind of the null model with regard to the interactive evolution of loci. Kind of like Hardy-Weinberg was our general population genetics null model. When we see a violation of linkage equilibrium, we know that there's something going on regarding how two or more loci are interacting in their evolution. And we call this linkage disequilibrium. Now, unfortunately, oftentimes when the term linkage is used, it's just referring to the latter, linkage disequilibrium. But if you just hear the term linkage, typically what people are referring to is linkage disequilibrium. And once we talk about some of the mechanisms associated with linkage disequilibrium, you'll understand why. All right, well, let's talk about the null model first, linkage equilibrium. Let's take a simple example where we're just talking about the potential evolutionary interaction between two loci. You got the A locus and the B locus, and they both have two alleles, the dominant and recessive in both cases. And if they're distributed among chromosomes in this way, we can count up the number of times that both of the dominants are together, different combinations of dominant heterozygotes, and what combination of chromosomes have both the recessives for the A locus and the B locus. So out of these 25 chromosomes, you can calculate the individual allele frequencies by dividing the presence of those divided by the total number of chromosomes. And so we end up seeing that we have 60% of big A alleles and 40% of little a alleles. For the dominant B, we have 80%, and the recessive B, we have 20%. Now, if we look at the way that those alleles are combined on a specific chromosome to produce combinations of A's and B's, we see that 48% of the chromosomes are big A, big B, 12% the combination in which you have a big A and a little b, and 32% are in the combination where you have a little a big B, and the rarest combination is when you have the recessive of both loci. Now graphically, this can look like this. Notice that over here we have the frequency of the B alleles. On the x-axis we have the frequency of the A alleles. And look at how organized this looks. Okay? So the largest number of A alleles was the big A, and the largest number of B alleles was the big B. So what would you expect to see the biggest combination of A's and B's? Well, clearly that would be the big A, big B. That's why this purple block is the biggest. And then given how common the big B's are, the remaining big B's are combined with the rarer A allele, the little a. Now you get down here, we talk about the little b. The little b is relatively rare, right? Remember, it was only at 20%. The biggest combination of those are going to be with the most common big A allele. And the rarest combination, again, is the rarest of both allele types combined. But notice how these lines line up here. This is indicating that each of the individual genotypes at locus A have an equal chance of having each genotype of locus B, given each of their relative frequencies. So again, because big A is the biggest and big B is the biggest frequency, that is the chromosome type that you should see more commonly. The rarest alleles, you would expect them to be rare, but everything is being represented in the proportions that you would expect given the relative frequency of the A and B alleles. So this population is in what we call linkage equilibrium. All combinations are being formed as you'd expected if they were just assorting at random. Now let's contrast this to this example. We still have 25 chromosomes, and if we look over here at the allele frequencies, they're the same. So that's not changing, but it's the combinations of these that are now changing. So notice it's easy, you can work through the math here, but it's easiest just to kind of look at what's going on up here to give you a good sense of this. This was by far the greatest combination that you would expect, the big A, big B, but actually it's underrepresented in this case. This over here is the combination of the remaining big B's, and they're overrepresented given the frequency of the little a. Yes, this should be a relatively rare combination, but it's even rarer than we would expect if these alleles at each gene locus were assorting randomly. When we see a pattern like this, again, you see these lines are not lining up here, this tells us that the evolution of alleles at one locus somehow are affecting the evolution of the alleles at the other locus. We see that they are somehow linked to each other. And this is why we call this linkage disequilibrium. Okay, here are our two models. Again, the one on the left here is representing our null model, linkage equilibrium, in which we have all possible combinations being shown in the relative frequencies we would expect if each of the alleles were assorting randomly. When we see a deviation from that, this null model of linkage equilibrium, we have what is called linkage disequilibrium. Something is occurring here where these alleles at the two different loci are interacting in ways to influence each other's evolution. The name linkage is derived, or linkage disequilibrium is derived by the physical linkage of gene loci that may explain this pattern in most cases. Because, I mean, if we think about it, genes are not floating around independently in the loci. Genes have a certain spatial arrangement on chunks of DNA, which we call chromosomes. And genes on the same piece of DNA, the same chromosome, are linked to each other to a greater or lesser degree. So that genes that are closer to each other, we say, are in tighter linkage. Genes that are closer to each other on the same chromosome, therefore, are likely to show a pattern of linkage disequilibrium. So let's look at an example. Let's say we have a parent that is heterozygous at the A locus and a B locus. Now, the possible genetic combinations they can produce in their gametes might be one genotype, A, B, both dominants, or they could produce both recessives at both the A and the B locus, or they could have a combination of dominant recessives at the A and the B. But what if they're more likely to produce just the dominant AB together in some gametes, and then the recessives on the other chromosomes that they form, the other gametes? Why are they not producing these other combinations? Because if you do your old Punnett square, you think that they should be able to produce all of these combinations. So graphically, this is what I'm saying. There are the possibilities of making all four of these combinations, but the boldness of these lines indicate the ones that they're more likely to be producing. Now, when we talk about allelic combinations in gametes, we are going to call these haplotypes. The reason for this is it's the haploid component, because remember, gametes are haploid in diploid species where the somatic tissue cells have two copies of each chromosome. Remember, the gametes just have one chromosome of each type, so that's the haplotype. And so at the A and the B locus, you just have one allele. And the allelic combinations in the haplotypes that just happen to be formed by this one individual, primarily it's going to be both the dominating the B or the recessive A and the B. Why are you not getting these other combinations? Well, here's a potential reason for this. It could be due to physical linkage of the A and the B locus on the same chromosome. This diploid individual that is the parent, on one of their chromosomes, at the A and the B locus, they have both of the dominant alleles. Then on the homologous chromosome, they have both of the recessive alleles. Now, if these gene loci are really next to each other, we say that they are spatially linked. The effect of this is it reduces the chance of recombination, or it reduces the chance of crossing over. Remember, in meiosis 1, one of the key factors that occurs is the homologous chromosomes to come together and they swap segments. This is recombination or crossing over. But the alleles at each of the loci that we're talking about here are not likely to be involved in crossing over, and so they're not assorting randomly. Here's a graphic representation of what I'm talking about. So we have two duplicated homologous chromosomes here. A crossing over is going to occur anywhere along this line, but if they happen to occur exactly where I'm indicating here, is this going to recombine combinations of A and B alleles? No, they're just being swapped in complete batches with the other chromatid. In contrast, if the A and the B allele were more widely spaced on the same chromosome, then there's a much higher chance that crossing over is going to occur somewhere in between the A and the B locus, in which case you would now get a combination of little a, little b, and big A, little b. You're more likely to get even representation of all four of these haploid types. Now, we use a shorthand for when we're trying to look at the potential haplotypes being formed. And so we do this by indicating which chromosomes they're distributed on. So the specific haplotype combinations. So the individual that I was introducing to you before by just the gene loci and whether they were homozygous or heterozygous, they were both heterozygous for A and B. But if you're going to rewrite this based upon the allelic combinations on each haplotype, then we would write it as big A, big B / little a, little b. So that would represent this situation. One chromosome has both of the big A and the big B. The other chromosome has the little a, little b. Now, when genes lie right next to each other, the probability that you're going to get crossing over is so rare that we sometimes refer to these as supergenes. They're basically acting as a single gene because crossing over really never recombines them. And sometimes this is a good thing. When you have certain allelic combinations at multiple loci that just really work well together, we call that a co-adapted gene complex. So specific alleles at each gene locus, they really work well when combined with each other, but not when they're combined with other alleles. All right, let's give an example of how linkage disequilibrium can be a mechanism to preserve a co-adapted gene complex. So in this example, I'm going to talk about 3 species of butterfly. 2 species are toxic. They're basically noxious so that if a predator eats them, it makes them sick. One species looks like this, the other species looks like this. So these are 2 different species. On the right-hand column here, we see that these are species that are mimicking the poisonous species. But this is a single species. Some morphs of this species mimic this butterfly. Some forms of this species mimic this species. And you can see that they're pretty good in mimicking these species in 2 important ways. It's going to be important to keep up with. First, notice the coloration pattern here. Pretty good match to that one. Coloration pattern here. Pretty good match to that one. Not perfect, but pretty good. But there's not only a color difference, there's a wing shape difference. This species is mimicking the tail of this species. This species doesn't have a tail because the species it's mimicking also doesn't have a tail. So there are two traits in broad generalization that make you a good mimic, or not, of the model species. The color pattern and the tail. The color pattern and not having the tail in this situation. So if you take this mimicking species into captivity, you tend to see that the offspring are going to be one of the two types, but you don't really get intermediates. So meaning if you get the color of the tail form, you also get the tail. If you get the color of the tailless form, well, they don't get a tail. So this kind of suggests that this really is a single trait controlled by a single gene locus, each of these morphs. So maybe morph 1, the tail morph you may get as long as you have a dominant allele, and you get the coloration and the tailless form only when you have homozygous recessive, for example. That could be a simple explanation for what's going on here. But it turns out that if you do breed these enough, rarely you get something like this. So in this situation, you've got the coloration pattern of the tailed form without a tail. So what's really going on here is it's not a single gene locus. There are multiple loci that are involved. Each specific component is driven by different loci. So that begs the question, why don't intermediates pop up more often? I mean, we should be getting different combinations of alleles at each of the loci for giving intermediate morphs. So let's say let's be simple here. There's a locus associated with a color, and then there's a locus associated with the tail. So why don't you get more combinations of the coloration pattern of having a tail without a tail, or having the coloration pattern of the tailless form that also get a tail with it? Because recombination through crossing over should be giving us all of these combinations. And also independent assortment could be playing a role if these loci are in different chromosomes. Well, the answer to this is these genes are linked. The gene loci involved in the formation of a tail or not a tail and the specific color pattern are really close to each other. They're spatially linked. So the mimetic genes are on the same chromosome and they're really close to each other, such that recombination really rarely gives you a combination that is breaking up the co-adapted gene complexes seen on each haplotype. So let's say that this is the combination that will get you the coloration of the tailless form and no tail. This is the combination that gives you the formation of, say, a tail of the B gene and the appropriate color for the tailed form. And it doesn't matter what happens, who you mate with, you're either going to get, the individual is going to combine this, pass on in their gametes this haplotype or this haplotype. You're not going to get combinations. So this linkage disequilibrium is preserving the co-adapted gene complexes. There are two co-adapted gene complexes in this case. You want to be one or the other, you just don't want to be an intermediate. Intermediates would occur if you had a situation like this. Here we see that there's less linkage, spatial linkage between the A and the B locus, and here you would expect to get more combinations of colored forms and tailed forms. So again, we have two regular combinations of alleles at different haplotypes. One combination has the tail and the appropriate color. The other one has the tailless form with that other color and pattern. And each of these combinations may have equal fitness in the population. So as long as there are enough model species of each type and they're being represented in those proportions, then they're great. They're modeling one species or the other, and they're tricking birds and other predators and not eating them because they think that they taste bad. So what would happen to those rare intermediates that pop up? Well, they're not going to be very good mimics, right? So they are not, that would be enough that a bird might say, huh, you kind of look like that toxic form, but something's not right there. Yeah, I'll take a bite and see. So there's not going to be selection pressure to preserve that. Those individuals are going to be taken out by natural selection. So what we're saying here is there's actually selection for having these haplotypes structured in a way that they're spatially linked, because linkage disequilibrium in this situation is preserving a co-adapted gene complex. So even if these haplotypes evolved without spatial linkage, there would be selection to drive a system where the loci are more closely spaced. Those individuals with greater linkage, that would help them preserve this co-adapted gene complex through the rearrangement of that chromosome, through mechanisms like we talked about in the past, genetic retroposition, in which these combinations are preserved. We'll talk about another mechanism a little bit later. So in this example, individuals with genes in closer positional proximity will have greater fitness. In this case, greater linkage disequilibrium is preserving the co-adapted gene complex, and so linkage disequilibrium is adaptive in this situation. Linkage keeps alleles that are appropriate together, even if individuals breed with another morph. They're not going to get inappropriate combinations of these alleles. So that's one scenario in which you might see linkage, but you can also see linkage in situations where genes are not spatially linked. So let's say that we have a short period of time where we have extreme selection preferring certain phenotypes over others. So certain allelic combinations may be preferred, and only those individuals survive or they have high fitness. Now, if there isn't spatial linkage, recombination will produce certain gametes that are not beneficial, but again, selection is just going to wipe those out before you could even measure that in the population, if those phenotypes have such extremely low survival. So we're going to have an overrepresentation of certain allelic combinations due simply to this extreme selection for those specific combinations. One potential example of that are the HLA loci that are associated with the production of a better immune system. There can be such selection for specific allelic combinations that those are preserved despite the fact that crossing over does produce intermediates occasionally that are just being weeded out by natural selection. And again, if an individual gets chromosomal rearrangement where these HLA loci become more closely spatially linked in the appropriate co-adapted gene complexes, then there's going to be selection to preserve those individuals that have this linkage disequilibrium that's due now to spatial linkage. Linkage in the short term can also occur due to drift acting on small populations. After a genetic bottleneck event, you may just see certain allelic combinations more represented than what you would expect. So this type of random event may just result in a generation, at least, having an uneven number of specific allelic combinations, either through, again, genetic bottleneck event or maybe a founder event. But if the loci are not spatially linked, we would expect after one generation or maybe a few more that this population is going to return to linkage equilibrium from a pattern of linkage disequilibrium. So it's going to be more likely a temporary situation here if there's no spatial linkage. So we're talking about the potential for linkage to change through time from generation to generation. How quickly does that occur? We're going to do just a little bit of math on this, and I'm going to give you a formula. D is a quantification of the amount of linkage between gene loci, and it can range from a positive 0.25 to a negative 0.25 when we have completely linked genes on either extreme. And we'll talk about these a little bit. It'll make a little more sense when I show you a figure here in a minute. This is these two numbers, 0.25 and a negative 0.25, have to deal with whether the haplotypes are segregating maybe all of the dominant alleles and the recessive alleles, or the opposite combinations of those. So knowing exactly why this is 0.25 and negative 0.25, don't worry about that right now. Just know that those extreme numbers represent completely linked genes, a complete pattern of linkage disequilibrium. Once we get to a D of 0, this represents genes that are associating gene loci where their alleles are associating totally at random. So all allelic combinations are seen as expected. So when we have D of 0, we're saying we have linkage equilibrium. So we can track changes in D through time, assuming Hardy-Weinberg conditions. So no selection, no genetic drift, no gene flow, random mating, no mutations. So what is going to control the pace of changes in D? Well, that's going to be all associated with the rate of recombination and the probability of recombination breaking up linked genes. What recombination does is it reduces linkage disequilibrium to a point of linkage equilibrium. The rate at which recombination will do this is dependent upon, again, the recombination rate, which is going to range from 0 to 0.5. All right, so let's explain the math associated with 0 to 0.5. Again, this is associated with crossing over, recombination, or potentially even independent assortment. When we have an R of 0, that means recombination is not going to occur between these loci. These haplotypes are going to be preserved. This is a situation where we're talking about like a supergene. The loci involved are smack dab next to each other. So the effective chance that crossing over is going to occur between them is nil. When we have an R of 0.5, then the loci that we're talking about are behaving as if they're on separate chromosomes. In this situation, we're going to have all allelic combinations having equal chance of forming. Now, why is this maximizing at 0.5? Well, this is because if they're on separate chromosomes, independent assortment is going to be the driving force here, determining recombination. And independent assortment is just random, right? So when the homologous chromosomes line up at the metaphase plate, they do so in a random order. And randomness is here defined as 50-50 chance. So the formula we're going to be tracking, the rate of change from linkage disequilibrium to linkage equilibrium is defined by this equation. D prime equals the quantity 1 minus R times D. D equals the present linkage level seen in the population at time X. D prime is the linkage in the following, the next generation. And once we get a D of 0, we have reached what we call complete linkage equilibrium. All right, let's just kind of understand what this formula represents. If we have an R of 0, where there's no recombination, this number is just going to be 1. So 1 times D, D and D prime aren't going to change. So we're not going to have any closer, we're not going to be changing our linkage disequilibrium from one generation to the next. Now, let's say that this is the opposite. It's 0.5. We're at 1 minus 0.5, that's half. So you take D, divide it by 2, or multiply it by 0.5, man, we're reducing the amount of linkage disequilibrium in one generation by a half. So think about the range at which R is going to affect D changing to D prime. And graphically, this is shown here. Again, if R is 0, and we start off with a coefficient of linkage disequilibrium being 0.25, it's not going to change. It's just going to go straight across. If R is even a little bit, a 1% chance of recombination occurring, it doesn't take many generations to drop a considerable amount. But when we have R at its maximum of 0.5, it really doesn't take many generations before you get to complete linkage equilibrium. And once we get to linkage equilibrium, it will be maintained until maybe selection prefers certain allelic combinations over others, or maybe temporarily associated with genetic drift. So it could shift us back to some degree of linkage disequilibrium. But again, R then will play its role and returning it back to linkage equilibrium depending on the spatial linkage between the associated loci. And here's a lab study that demonstrates this. So they took a population of flies with two different loci, and they took populations that are fully in linkage disequilibrium for one extreme or the other. So one population had the haplotype big A, big B. The other haplotype at these flies, their other chromosome, was the haplotype little a, little b. Okay, and there's another way that you can measure linkage disequilibrium from negative 0.25 to 0.25, and it's just scaling it to 1. So don't let that confuse you, but notice it's 1 to negative 1. Okay, it's just when you have this, we're talking about complete linkage disequilibrium for this combination. Here we have this combination being represented by a complete linkage disequilibrium. It's just a different combination of haplotypes. Regardless, what we can do is see how well our predicted rate of going from this extreme linkage disequilibrium, how it's going to approach, how quickly is it going to approach linkage equilibrium. This predicted line is associated with the formula I just gave you. Here are two different experiments, with different experimental populations starting at either this level of linkage disequilibrium or this level of linkage disequilibrium. The specific pattern varies from trial to trial, but they're all showing a pretty good match to this prediction. And again, why is there such jaggedness associated with these lines? And why do they differ somewhat? That's simply due to genetic drift. That's genetic drift because we're dealing with relatively small populations of flies in each of these studies. And so you're going to see generation to generation shift in these haplotype combinations due to genetic drift. But this is a pretty powerful demonstration that recombination, even for populations that are completely in linkage disequilibrium, in just a few generations, so about 30 generations, they're already getting really close to linkage equilibrium, and by the time we get here, most of them are, except again, sometimes we get genetic drift causing some bizarre deviations. Most of my previous examples of linkage disequilibrium have involved loci that various combinations of the alleles at those loci make a co-adapted gene complex. We can actually find other cases of linkage disequilibrium that's not necessarily adaptive. So, for example, sometimes neutral or even slightly deleterious alleles can hitchhike with a selectively advantageous allele, which this forms a pattern of linkage disequilibrium, but it's not necessarily that the associated loci have specific combinations of adaptive alleles. Let's talk about a situation where linkage disequilibrium is actually neutral from a perspective of adaptive evolution. In this case, a neutral allele may be hitchhiking with a selectively advantageous allele. So let's talk about how this would come about. First, let's talk about how selection might favor a new mutant in a population, such that at locus A, before this mutant arrived, this was associated with some fitness. Individuals with the A locus had some fitness. But if we get a new favorable mutation, the A prime, it may be associated with an adaptive response such that the bearers of the A prime allele, this new mutant, have significantly greater fitness. Well, as we've mentioned, not all of the gene loci are assorting randomly. They're not all floating around independently in the genome. They can potentially be linked to each other on the same chromosome. There is some gene locus that's close to the A locus. Let's say that that's the B locus. And at this B locus, there is a two allele system in existence. So you either have the B or the B prime. But really, there's no selective difference between them. Each of the B alleles has equal fitness. Such that at the A locus, the individuals that had the original A allele, just the plain A allele, sometimes they were combined with the B allele at the B locus, and sometimes they were associated with the B prime allele. But when we get this new favorable mutation in the population, it has to be linked to just one of these alleles, and if it just happens to be associated with the B instead of the B prime, well, the B allele now is going to become much more common in the population. Is this because the B allele is better than the B prime allele? No, it has nothing to do with that. Remember, they are neutral relative to each other. But the B allele is hitchhiking on the success of the A prime allele just because it's very closely spatially linked. Now, the degree to which this association of the A prime and the B, creating a pattern of linkage disequilibrium, the time in which we see this pattern of linkage disequilibrium depends on how closely they are spatially linked, because recombination is eventually going to break this up. Again, that's just go back to what we were just talking about. The D prime equals 1 minus R times D. So the magnitude of R is going to determine the length of the preservation of this pattern of linkage disequilibrium among these allelic combinations at these two loci where there's really not an adaptive response. However, in some cases, we can get a pattern of linkage disequilibrium that is actually disadvantageous. So I'm going to give you a couple examples of this. Let's say that we get a new mutation that is a good mutation, that it in itself could give the bearer of this new mutant beneficial fitness. But if it's linked to an allele at another locus that's working in a different way, it may limit the ability of this new mutant to fully increase the fitness of the bearer of this new mutation. So in this case, it might slow down the rate at which this mutation could spread in the population. If this is the case, though, eventually this mutation could spread quickly if the linkage disequilibrium is broken up by recombination. All right, so let's give an example of this. We talked about alcohol dehydrogenase. This gene locus is associated with different capabilities in fruit flies with regard to digestion of ethanol, alcohol. And there are two alleles. There's an S allele and an F allele. The S allele is associated with really not being able to utilize alcohol. And let's say we start off with a population in which this is the only allele in a population. All of the individuals are ADH-S. Now, some of these flies in this population have a natural aversion to alcohol, behavioral response. Others have an affinity to alcohol. Let's then say that in this population, we do see the evolution of a mutant. We see the evolution of the ADH-F allele. If this mutation happens to occur in an individual that has alcohol aversion, that's just a tragic situation, right? Now they have the ability to utilize this alcohol as a food source, but they have a natural behavioral aversion to this so that they won't even try it. So here we have the potential beneficial mutation, but because it's in linkage disequilibrium, close spatial linkage with the alcohol aversion allele, we're going to see the potential for this beneficial mutation to spread in the population is going to be limited. So this is limiting the potential for this new mutation to spread in the population. Once we break this pattern of linkage disequilibrium, however, and we produce individuals that have this combination of alleles at these two loci, the alcohol affinity and the ADH-F allele, then we can see this combined trait sweeping to fixation potentially in the population. Let's look at another example. So we may have a positive mutation that's under directional selection, but if it's linked to a locus in which you only get an adaptive morphology when you have the heterozygous condition under stabilizing selection, well, selection can't work in both ways at the same time. Selection will try to drive the fixation of the new mutant in the first example, but if it does this, then the allele at the second site will be dragged along too that just happens to be linked with it, and it won't be able to form its heterozygous combinations anymore. So one of two things is going to happen. Whichever is more important from a selective point of view may drive the bus relative to maintaining heterozygosity in the population or going to fixation at the other gene locus, or if recombination can break these apart where you can find different combinations, then you can have selection operating independently on each of these gene loci. But at least the initial linkage disequilibrium is going to be disadvantageous. So just in general, how common is linkage disequilibrium? Well, it turns out most loci are spaced far enough on a chromosome that they can be pretty close to linkage equilibrium. I mean, even within, because most loci are going to be on different chromosomes. Okay, and independent assortment is going to take care of it. But even within a chromosome, especially if they're not next to each other, we tend to be at a state of linkage equilibrium. And this really just goes to prove the importance of sexual recombination, how powerful it is, and that's going to be the topic of the next lecture. So this is just data from human chromosome number 2, showing you that once we get over, about 100 kilobases in length between loci, we're pretty much at linkage equilibrium. So what's the point of this then? What linkage can occur? We've talked about how it can occur and how it can be reduced. What are the mechanisms associated with it? And actually our understanding of those patterns can be used as a tool for understanding other aspects of evolution. So for example, the time of the origin of allele in a population and when it first becomes linked to another gene locus. So that's what's being shown in this figure here. We see that if we know what the present linkage disequilibrium is between two loci, and we know the pattern of linkage disequilibrium decay from that formula that we talked about, that we know how that changes from generation to generation, we can scale that to figure out, first, what was the degree of linkage disequilibrium at its origin, and how many generations has it been since that linkage disequilibrium occurred. So that's assuming that the decay of linkage disequilibrium is not being influenced by something by selection. But if this curve is deviating from our expectations based upon recombination being the only factor, then we also might have evidence for selection operating and the strength of that selection as it might change the relative curve or position of this decay line. Now that was a quick review of kind of how we can use linkage as a tool. And if you want more details of this, see these pages in your textbook. Okay, so I understand that linkage disequilibrium can be a fairly difficult concept to get a good grasp on. So you may have to watch this video a couple of times, or you may have some questions for me. I'll be glad to answer those. The next topic is going to be contrasting the evolutionary significance of sexual reproduction versus asexual reproduction.